that's exactly right. I'm a genetic counselor at Boston Children's Hospital. I'm one of the curators as well as the coordinator for the brain malformation expert panel in both the GCEP and the VCEP portion. Sorry, my Zoom is blocking off half my screen, so I'm gonna fix that. Okay. And just a quick background about the expert panel. These are our current members as they stand now. We started in 2018, and we've been working on gene curations and variant curations since then. Our experts and curators come from all around the world, encompassing many aspects of brain malformations. We have individuals who see the patients like myself and many of the neurologists and geneticists on our team. We also have a lot of the researchers who do the bench work and have published on these genes. And we also have some collaborations with clinical laboratories who are doing somatic testing, and I'll talk a little bit about that kind of at the end. But we are a pretty multi-nationality group. We have people from all over the world. And a little bit of background about the 4 genes in particular. So MTOR AKT3, PIK3CA, and PIK3R2 are all within the MTOR pathway, which is like a very well-known pathway in many different biological processes, including some cancer stuff. So maybe this might be a prelude for the next two meetings. But our VCEP is focused on somatic variation in these 4 genes that result in a brain malformation phenotype. We recognize that there are many different kind of special considerations for these particular genes, and do recognize that there are non-neurological phenotypes that arise as well. And so, we've come up with a name, which is pretty long for the disorder that these genes cause, and it's overgrowth syndrome and/or cerebral malformation due to abnormalities in the mTOR pathway. And this name applies to all 4 of them. This is some of the work that we've done in the GCEP actually, but if you look in OMIM, you'll see, particularly for PIK3CA, there's like 12 disease entities in there, and a lot of them are overlapping. And so this is the name that we've come up with. When we're thinking about these particular genes, and particularly somatic variation within these genes, there are special kind of curatorial considerations that we've had and had to think a lot about. One of the main points is which of the ACMG criteria account for post-zygotic change versus germline mutation? Are there kind of considerations that you need to think about? The disease mechanism for these 4 genes that results in the overgrowth is a gain-of-function disease mechanism versus the loss-of-function disease mechanism in some of the other genes in this pathway, like P10 or TSC, the tuberous sclerosis complex genes. And with gain-of-function variation, functional validation is very important, and exactly what constitutes good functional assays in this field. And so, I'll be talking about all of the amendments we've made to the ACMG criteria, and I'll highlight a number of them that we've, added a lot of specification for to address some of those considerations. So to start off with, the combination rules that we've adopted are out of the ClinGen group, that Bayesian kind of numerical system. We have compared this to the traditional original ACMG, combination point-based system. And for our group, we really like the numerical system better. And that's because we do a lot of curation actually outside of the VCI. We have a lot of kind of sort of mini scoring systems that I'll talk about, but by doing it outside and having a numerical system, it actually makes it a lot easier to program the Excel spreadsheet that we use to generate the classifications. And also there has been debate in our VCEP about how to weight, the pathogenic versus benign criteria if a particular variant meets criteria for likely benign, but does have some pathogenic points as well. How do we deal with that? And having a numerical system, which is more concrete, has been very helpful for us. So just as an overview of the adjustments that we've made, of the original 27 ACMG criteria, 4 remain unchanged. So 23 of them, we have added some type of specification to, either modifying the strength or adding some type of gene or variant specific clause to it. We validated with 43 variants across the 4 genes. Of the 17 pathogenic and likely pathogenic variants, 4 of them were not in ClinVar, and 8 of the 13 that are in ClinVar had 0 stars. There are 2 in ClinVar that had no disease assertion. And you may be thinking 17, that's actually not that much, but actually for this VCEP, that's actually like over 90% of the known pathogenic variants in these genes, because a lot of gain of function variants have very specific loci that are affected. They're all kind of hotspots, and so they're very highly recurrent. And so that's, I know they had recommended like 10 benign, 10 likely path, and all of like 10 of each classification for each gene, but because ours are so specific, we have a little bit less than that. Okay, and so I'm going to be going through the criteria now, and I'll start off with the very strong pathogenic points. And so PVS1 doesn't apply to us because our disease mechanism is gain of function, not loss of function. PVS4, the way we had looked at PVS4 was borrowed from the P10 working group. We really liked how they added a lot of specificity to the various phenotypes that are associated with P10, and kind of modulated the strength based on the number of probands and how specific a phenotype that proband had was. And so that's represented in this table. This is in the spreadsheet that I was talking about that we use to curate our variants. All of this is built in, so the curators just have to identify what phenotype the proband has, and it's a drop down. So each individual that has neuropath confirmed focal cortical dysplasia, for example, will count for 1 point, and each of those probands, as long as the curator lists how many probands there are that meet that criteria, then the proposed score and PS4 strength is automatically calculated for them. In PS4, we did have a couple ways in which we addressed some of the curatorial considerations that I had mentioned earlier. For the post-zygotic versus germline, comment, we recognize that the phenotypes for these disorders are actually very diverse, and there's kind of a couple reasons for that. One of the main reasons is somatic variation can occur in basically any tissue, and depending on the tissue type that is affected, you will have a different phenotype. And so we wanted to encompass all of those phenotypes, not just the neurological ones that are obviously the brain malformation group is most interested in, however, also the non-neurological tissues. So if you had one of like a PIK3CA pathogenic variant in a limb or just the hand, some of these individuals will present with non-symmetric overgrowth or macrodactyly of a single hand. And so, we did want to encompass a wide range of phenotypes for this. Another thing that is used a lot in some of the older papers that first reported on somatic variation in these genes causing a congenital disorder is the use of the presence of the variant in tumor samples in databases like Cosmic. And so we did want to include that information as well. We did set a couple limitations to that though, and we believe that this information is really important. However, it should not be used at a strength above supporting. And so if a variant only has tumor-specific data, no matter how many tumors a particular variant is present in, we feel that it should not be weighted above supporting. So even if a particular variant is found in 300 tumor cases, as long as there's no proband with a neurological phenotype or congenital phenotype, then that PS4 will remain at supporting. Additionally, no more than 9 tumor samples can be accounted for. And the reason for that is keeping in line with the idea that we didn't want the tumors to be weighted more than supporting. And the idea is that if you had a single proband that met a proposed score of 1, even if you had 3,000 tumors, we didn't want one proband and 3,000 tumors to result in a PS4 very strong criteria. And so we set the limit at 9 so that even if you had one proband, it wouldn't rise above a moderate score. Additionally, one of the addressing some of the functional considerations was, can you guys see my pointer, my mouse?
I cannot see it. Oh yeah, there you go. Yeah, you can see it now. Yeah, there you go.
So there's this line with 4 asterisks, essentially saying, functional assays using patient-derived cell lines or patient tissues that show a cell growth phenotype or increased phosphorylation now is instead of being used as PS3, which is the functional criteria, it's added to this PS4 criteria. And the idea is that the individual already has the phenotype, so you can expect that the cellular pathology or the functional assay using their tissue is going to recapitulate that. And that's more of a phenotype rather than a functional assay. And each variant can only have this criteria applied one time. This is probably the most common functional assay that is used in these 4 genes. A lot of the older papers from 2012, 2013 basically take tissue from affected patients and do the staining, do the phosphorylation, and that has been kind of a very, very used motif, and a lot of different researchers actually use the same system to test, and but they're all doing the same thing. And so we felt that we wouldn't want to double count all of those functional assays because they're all essentially testing the same phenomenon for the same variant.
Abbe, are you okay with taking questions throughout the presentation, or would you prefer to wait until the end?
Sure.
Okay, there's a question in the chat. Can you elaborate on why variants found in tumor samples can be used as evidence of PS4?
Oh, yeah. So that's a good question. The idea is that particular variants in tumor samples, the reason that those variants occur is because they offer some type of growth advantage to the cell, theoretically. And so actually a lot of the variants that we are looking at are strongly activating in the MTOR pathway. And so they're in many thousands of tumor samples. And theoretically, the focal cortical dysplasias and the hemimegs and the overgrowths share that kind of overgrowth theme with cancers. Individuals see focal cortical dysplasias and have described them as benign tumors, essentially. And so that's the reason that we use the tumor samples. Does that make sense?
Great, yeah, thanks. And I have a quick question as well. I love this point system, and we've actually been working on some of these sort of point systems for PP4 for some of the metabolic disorders as well. But I wanted to ask you, how did you come up with the different numbers of points? Is this based on expert opinion, or have you done any sort of statistical analysis to provide evidence for these numbers of points?
Yeah, so this does come from the P10 group. I'm not actually sure exactly how they determined the thresholds, but when we did present this to the SVI committee, they had recommendations on what they wanted the points to be, and that was based on some statistical analysis. Originally, our very strong point was at 7 points, and that didn't fit in with the statistical analysis that they had done, and so we had raised that to 16. And the idea behind the proposed score is there are certain, it's basically how specific the phenotype is to these variants. You'll see if you have neuropathology confirmation, that's weight a little bit more than if you didn't have neuropathology confirmation, and that's because some of these very subtle MRI findings on pathology can actually not be like an FCD. They can be like a tuber or something else, or normal cortex.
Thanks.
So now going on to the strong criteria, we left PS1 the same. PS2, we did add a little bit of specificity, again, kind of addressing the somatic, the post-zygotic nature of our variants. So we split PS2 into 2 separate criterion. Criteria 1 is your traditional de novo with confirmed maternity and paternity. The second criteria is a particular variant present at an increased allelic fraction in an affected tissue versus an unaffected tissue. And that is sort of demonstrating that this particular change is a post-zygotic change. And if only criteria 1 or criteria 2 is met, then a PS2 moderate criteria can be applied. If both are met, then a strong criteria can be applied. And we do recognize that as new technologies arise that are able to detect variation at even lower and lower allele fractions at like 1% or less than 1%, having a way to really validate that is going to be very important. We do strongly encourage having an orthogonal confirmation method when you are evaluating for criteria 2, because if something is present at 0.5%, it's you have to make the argument, you have to make us believe that this is not a sequencing artifact. Okay, and then for, I'm not going to talk about PS4 again, that accounted for the very strong, moderate, and supporting already, but for PS3, we did work very closely with the SVI committee to basically refine the basic quality metrics for certain assays that are used in our 4 genes. This does not apply to animal models, though. If an animal model is generated and with a variant expressed in neuronal tissues and shows a good neuronal phenotype, then that still can be awarded with PS3 strong. If an animal model is generated that shows like increased cancer burden or increased cell proliferation, then that is a PS3 supporting or a moderate point. But I will kind of show you what the validation for PS3 looked like for us. And I think I saw Sharon and Donna on the participation list, but the PS3 criteria for us was modified from the Brnich et al. Paper out of ClinGen, and we worked very closely with Donna and Sharon on how to do that, basically establishing what the minimum criteria are in order for a particular functional assay to be scored at supporting moderate or supporting moderate or strong for benign and pathogenic. And so I'll kind of describe the way we did that and what the quality metrics are. And the only thing that I am going to mention on this slide is basically in order to use a functional assay at a level of moderate or strong, you need a lot of validation variants, which are variants that are pathogenic or benign without the use of PS3 or BS3. Our variants are never ever going to be able to meet the criteria that is set. because again, our cases are highly recurrent. You know, there aren't, many of the genes don't even have 8 known pathogenic variants because they're all kind of hotspots. And this is something that, you know, I've discussed with our team, as well as, you know, the SBI committee, on ways to kind of address this. One of the theoretical ways is, you know, there are a lot of individuals who do good functional assays on these genes, and obviously it's the same variant, but counting each assay as a you know, sort of validation control. So if there are like 8 publications that show the same phenotype, or show the same you know, assay results, that may count. That's something that we are going to, you know, refine hopefully in the future. But these are what the minimum criteria we established were. I'm not going to kind of read these out, but essentially the idea is that for a given functional, like, class of functional assay, there should be just a basic, like, minimum standard that is applied, so that you can be sure that the particular functional assay is doing, you know, what it's promising to do, and it's not just you know, the result is not the result is reliable. And the way we did that, this is just an excerpt from the pretty large Excel document that we have with all of our functional assays. Luckily for us, for our 4 genes, there were not that many functional assays that we had to look through. There's about 20 in total that we assayed, and the general classes of assays that are used in our field are phosphorylation, depth-door binding, cell survivability, and cell proliferation. The names are pretty self-explanatory, but we basically got all of the functional assays together and analyzed them as a cohort, analyzed all of the different assays that looked at phosphorylation in particular cell types, and saw what features they had in common. Were there certain criteria that was not necessary? And a lot of times, actually, some of the publications didn't include that information in the actual report, but we said it was okay if you reached out to the researcher and they said, yes, we did do technical replicates, and they were done in 3 technical replicates, then you could just document that, and it would meet the criteria. Luckily for us, a lot of the individuals who were doing the functional assays are already on our VCEP, and so asking them wasn't very challenging because they were already there. Okay. And so that's all I'm going to say about PS3 and BS3. I'm happy to talk more about exactly, you know, how we did it, but it did take a good amount of time. And again, thank you, Shannon and Donna, for bearing with us as we were, you know, bumbling through this. And onto the moderate criteria, PM1 and PM2 were, you know, downgraded to supporting, and so I'll talk about those in a little bit. But PM3 was not applicable for us because ours are not recessive. Protein length changes for PM4, we also decided not to include because there's one known indel in PIK3CA that results in a gain of function phenotype, and it is fairly recurrent. It's one of our likely pathogenic variants. However, most of you know, the other variants in these genes are missense changes, and it's very difficult to, just based on looking at a particular variant, to know if a gain of function phenotype is expected from that change. Just because if you have 2 nucleotides right next to each other, one may be the highly recurrent one, but the other one right next to it might actually not do anything. So just based on the protein length change, you know, we decided not to include that, even knowing that there is one out there. PM5 we left by itself, and PM6 is addressed in our PS2 criteria. And then now for our supporting criteria, PP1 is not applicable. There is not going to be co-segregation in multiple affecteds. To our knowledge, there is no affected individual that has affected children. There are reported cases of germline mosaicism in this cohort, but that is extremely rare. For PP2, the constraint Z-score cutoff that we are using is 3.09. Interestingly, all 4 genes used to meet criteria for this. However, with the new iteration of Nomad, turns out PIK3R2 has a Z-score cutoff of less than 3. It's like 2.2 or something like that. And so I did have to go back and change that. For PP3, we also opted not to use in silico prediction tools in our curations, because same type of idea as the indel criteria. It's very difficult to assess just based on the variant alone if the particular change is going to result in a gain of function or a loss of function. And a lot of the in silico tools we know are based on the idea of like damaging or deleteriousness, based on the chemical differences or the conservation of a particular nucleotide or amino acid. And although it's true that the conservation of a lot of these hotspot variants are highly conserved, it's very difficult to determine which variant is going to result in a gain of function without functional validation, without true functional validation. And so we opted not to use the in silico tools. And PP4 is accounted for in PS4, and no one in ClinGen is allowed to use PP5. And as I had mentioned, we had downgraded PM1 and PM2. The reason we had downgraded PM1 is because the actual functional domains that are important for these genes are actually quite large. They're sometimes hundreds of amino acids long and span a good amount of distance in the gene. And the reason we opted to do it this way instead of having individual hotspot variants is because the actual recurrent nature of a lot of these genes we have captured by the PS4 points if a particular variant is in a lot of people. Because it's a hotspot, then you're already counting that for PS4. And so PM1 is really just telling us, hey, is this in an important functional domain of the protein? If yes, then it is a supporting point, because again, it's very hard to tell exactly which part of the functional domain is going to give you that gain of function effect, because that effect seems to be very, one amino acid or 2 amino acid dependent. And so that is all of the pathogenic criteria. Before I go on to the benign criteria, did anybody have any questions about any of that?
Abbe, I have a quick question about PP4 and PS4. So as you said, you don't count PP4 because those cases would be counted under PS4, is that right? Mm-hmm. Okay, great. I'm asking this because this question actually came up on a call last week, and none of the groups I work with use PS4, so I was just curious about that. So presumably when you are recording any details about those cases in the VCI, you'll be doing that under the PS4 code, just under PMID and so on. Is that correct? Okay, great. So hopefully that's helpful. I don't know if the folks who asked that question are on this call, but thanks for clarification, Abbe.
Yes. One other thing for PS4 is we specifically also have the caveat that in order to utilize that criteria, the particular variant does have to be absent or near absent from controls. Because again, a lot of these genes are highly present in cancers, and so we know that there are cancer cohorts in Nomad, and so we definitely don't want to be counting potentially benign cases and counting those probands as well.
And there's a question in the chat. Was cancerhotspots.org considered for hotspot determination?
It wasn't. I'm not familiar with that that tool, actually.
Yes, I'm not either. I'm not sure who asked this question, but if there's any other information about that, we'd certainly be interested. And thanks for bringing up the question.
Definitely. There is another website that I have used. Actually, it's not available for all of the genes, but it is available for PIK3CA, and I can't remember the name of it. Once I stop sharing, I'll find it. I bookmarked it, but it shows like all of the variants that have been reported in the literature for a particular gene, and like gives you the PMIDs for publications that are like in there, and gives you a little excerpt about if this is you know, damaging or not based on the functional assays that was done in the paper. And I thought that was pretty cool.
Yeah, sounds great. Thanks. And I see it was Dimitri who asked the question, so thanks for bringing up that website too. We can certainly look into that one as well. Thanks.
Okay. So, you know, the first step of our benign criteria was determining what the accepted allele frequency, or the maximum allele frequency, is going to be. These are very rare disorders, and so there is not a good estimate of the disease prevalence in order to calculate the maximum allele frequency. And so this is the math that we had done based on a whole host of publications, some of which are admittedly a little outdated now, but the rationale is about 1% of the population has epilepsy. 1/3 of that population will have refractory epilepsy or medication-resistant epilepsy. And of those individuals who have medication-resistant epilepsy, 1/3 will go to have surgery and be found to have a cortical dysplasia. And no single gene accounts for more than 1/3 of all of the causes of cortical dysplasias. And we used 1/3 as a very conservative estimate, because the more common genes are DEPDC5 and some of the loss of function genes like TSC1 and 2, not so much MTOR or any of the other genes that we're looking at. But the idea is we wanted to kind of create a set list that could be used for basically any variation in the mTOR pathway. And so we wanted to keep it pretty we didn't want to keep it as strict. I can't think of the word for less strict. Okay, and so for BS2, the particular variant is observed in healthy adults. We encourage at least 3 or more well-phenotyped family members be identified before applying this point. However, we also included homozygous instances in NOMAD to count for this. And so if there are 3 or more homozygous instances in NOMAD, then BS2 can be applied. BS3 was addressed when I had talked about the PS2 criteria, and BS4 is not applicable because there is not going to be segregation in affected family members. To date, there has been, as far as we know, there is no affected individual having affected children. BP1 is not applicable because our disease mechanism is not loss of function. We have kept BP2 and BP5 the same. These genes do not have repetitive regions in the exonic regions, and so that is not applicable. For BP4 and BP7, these criteria are only applicable to like non-coding or synonymous variation. Again, we didn't want to use the computational evidence for missense changes, but for splicing changes, which are most often associated with a loss of function phenotype, we felt that would be appropriate. And so 2 out of the 3 splice prediction tools must say that it does not have a splicing effect in order to award BP4. And if there is no splicing effect, then we look at the nucleotide conservation for BP7. And if the nucleotide is non-conserved, this point can be awarded. We have defined conserved as the same nucleotide present in all vertebrae. And that information comes from, that we use comes from either Alamute or the genome browser. And so that is it. We have submitted our criteria and have received feedback and have submitted the feedback, submitted edits based on the feedback as of the end of last month. And so we are very hopeful, very, very hopeful.
Well, best of luck, Abbe. That was a really great presentation and just really interesting to learn about how your VCEP has handled some of these issues with both the somatic and the germline variants. So yeah, it was really, really great work. I was curious about your PS3 criterion. So you had mentioned that you know, because these variants are so rare, there really weren't very many benign and pathogenic controls. And the VCEPs I work with are typically the metabolic ones. We've certainly come across this issue as well, not because the variants are rare, but just that we don't have a lot of controls. And so I was just curious, so you're using this criterion at strong. Was that for just one particular assay, or did the variant have to show that it was deleterious across a range of assays in order to assign PS3 at the strong level of evidence?
So none of the assays that we had looked at were able to be used at a strength above supporting. And so the PS3 strong, the only one that we have is the animal model, which doesn't, which isn't utilizing that. And so that is an issue, and I hear that is not unique to us. A lot of the VCEPs are also having trouble finding validation controls for particular studies. And that sort of makes sense. I mean, a lot of people don't use like grant money to do an assay on something that they think is already pathogenic or benign. But
yeah, that's true. Well, thanks for the clarification. Yeah, I just wasn't sure what strength you were using that at, but that makes sense. So thanks for that clarification.
We are hoping though that it's possible that if multiple functional assays are across different institutions are showing the same functional assay, that you can count those as like validations. And so if you have 8 assays that show the same thing, those assays can be used at a moderate level. I don't know if that would work, but that is something that we have thought about.
Yeah, and we've thought about that too. So it's kind of interesting because I suppose, yeah, it's sort of validating the, I guess, kind of the experiment in a way. Yeah, we haven't really come up with an answer for this one either, or whether there just may be some element of the experiment that's just causing that variant to look like it's deleterious when it just may not be, even though the experimental protocol is sound. So I guess that's been one sort of bit of feedback that we got. It could look like it's deleterious across these different assays, but maybe it really isn't in vivo. So that was one piece of feedback that we got.
Yeah.
So interesting to see how this moves forward. So I did see, so Dimitri had mentioned this website. So Dimitri, did you want to say a few words about cancerhotspots.org?
Can you hear me?
Yes.
So I put the cancerhotspot.org in the chat so everyone can see it. So this is the good resource because it takes in account the mutability not only at the gene level, but also at the particular nucleotide context. And as some people may remember, there is actually now the criteria which was approved by ClinGen CVI, which takes in account the somatic hotspots. And in particular, it uses the cancerhotspot.org. Yeah, so this is the resource, and there is the information how it was generated. One important thing to keep in mind, the statistical significance, it's calculated at the position and not at a particular amino acid change. So this is an important thing to keep in mind. And sorry for background noise, there is construction just outside of my condo.
No, you're fine. No, this is great. Thanks so much.
Cool.
And Abbe, I saw that you had shared your resource as well in the chat. So thanks.
Yeah, this is the resource that I had briefly mentioned. It's called the Clinical Knowledge Base from the Jackson Lab. The goal is interpreting cancer profiles, and so it doesn't have a lot of genes, but for PIK3CA, it does have a lot of, I can just share my screen so you guys can see what I'm talking about. The variants that are in publications and kind of the effect of the protein based on the publication. And this was pretty good. This was a good resource for us to find the functional assays that we used.
Great, thanks. And I see that G has shared a resource as well, a paper integrating somatic variant data for biomarkers for germline variant classification and cancer predisposition genes. Thanks, G.
Yeah, this is just a paper from the ClinGen somatic germline working group where proposed the cancer hotspot.org website.
Any other questions for Abbe?